This is a production of Cornell University.
Good afternoon. Thanks everybody for coming. And today we're going to talk about sugarcane. We're also going to talk about genome prediction, which is kind of your topic here, but maybe sugarcane is something that you hear about every year. So for those of us that didn't see sugarcane in the field yet, that's sugarcane. So sugarcane. Sugarcane, as you might know, is used for producing sugar. And also another very common use of sugarcane is to produce ethanol, and both for human consumption and for cars consumption. And that happens especially in Brazil, which is the largest producer of sugarcane in the world. And actually, sugarcane is grown in areas of the world. In Brazil, the top producer, followed by India and China. And despite not being among the 10 most planted crop in the world in terms of area, it's the one that produces the most in terms of total production. And just more interesting facts about sugarcane. So it's a plant from the grass. It's also a C4 plant. It's a high polyploid species, so it's actually the varieties, the clones that we see in the field today, they are hybrids from two polyploid species, Saccharum officinarum and Saccharum spontaneum. It's also outcrossing, and it's a clonally propagated crop. So it's going to have some impacts on how we manage the breeding program. And also, it's a perennial crop, so harvest happens every year after a growing season, and then growers can get average of 5 harvests from each crop. So why to do genome selection in sugarcane? So genome selection is being a tool that is working very well with quantitative traits, and those are the ones that are interesting in sugarcane. So we've been, also sugarcane has really long cycles, especially when you consider the cycle since the time that we do a cross until the time that we can do a cross again. It can be pretty long. And actually, the traits that are interesting are also quantitative. That's why we went on to study how can we, or how good can genome selection works for sugarcane. So just an overview of my presentation, I'm going to start describing my data sets, both the molecular markers and the field data, and then present the 3 main results that I have, which are prediction for clones, prediction for parents, and then another way of looking to the molecular markers in sugarcane. So field data. So I'm going to give an overview of the breeding program in sugarcane in broad terms. So you can split the breeding program in some selection stages. We start with the first stage, which is actually called a family stage, where families are planted together and evaluated together for traits like bricks and weight. Then those families that are selected, the clones that are the plants that are selected are clonally propagated, and then we start having stages. The initial plants are not only propagated, but then with larger plots, we can have better phenotype of the plants. So this happens normally around 2 years to move from 1 year to move from one stage to the other. When we get to the third stage, when we start having a bigger plots where we can have measurements that we consider important for selection, like sucrose and weight. And we can see the performance of the varieties in a better resolution. At the same time, here is still normally on a single location. And then on the following stage, then there's further multiplication where plants can be evaluated in different locations and actually through many more years. So the data that I'm using here comes from CTC, as Mark mentioned, which is the Sugarcane Technology Center in Brazil. Here's translation. Here's the overview of the headquarters and also our breeding station that stays here in the north of Brazil. Those are some growing areas in Brazil, the main growing areas. So let me go a little more in details about my data set. So I have data from 2 breeding cycles, what I'm calling cycle 5 and 06. They follow the same scheme that I described before. But on the 3rd stage, we had the actual phenotyping that I'm going to describe on the first, on the following chapter, and then use on the following ones. So on the 3rd stage, we got data that was on all these traits: bricks, pole, fiber, and weight, and also sequencing data, from which we got SNP information. So basically, the SNP, the molecular marker information that we have come from the 3rd stage, and we have 2 cycles to do our analysis. So just another statistics from this data set. Here I'm showing the size of the families in this 3rd stage, and we see that we have some families that are pretty large, but most of them are small. So there will be some unbalance in terms of the families on the 3rd stage due to selection. So let's take a look at the molecular marker data. So we did sequencing for genotyping from about 2,000 samples. We used a technology that uses some probes to capture the locations in the genome that we are actually getting our sequencing from. And then from that, we got a raw data set of more than 200,000 SNPs that later was further filtered out. So we got to a final data set with 55,000 markers in more than 1,700 clones. A bit more on the molecular marker information. So here in my initial analysis, I'm gonna consider the SNP information in terms of the presence of or absence of the alleles. So basically, I'm going to use a simple representation where I get a distinction between the heterozygous state from the homozygous state. Only that. We're going to talk more about it at the last section of the presentation. So with the marker information, we did relationship, we estimated relationships from the clones, and here I'm going to show a broad view of the relationship between the individuals based on a principal component analysis. Here, actually, those 3 plots are the same. It's just the colors that are different. And what I'm showing here in the colors are first here the cycles where the clones came from. So basically, I'm showing that there's no particular difference between the clones from the 2 breeding cycles. On the other hand, when I start putting colors based on which female parent or male parent the clone came from, we start to see a relation between the colors and the dots. So basically, the structure that we can see in the among those clones is more related to the parents that they came from than from the cycles. And that kind of makes sense when you see how the parents were used in the different cycles. We have some overlap between parents, and also even when the parents don't overlap, they are related. Move on. So we also have pedigree information from those clones that we use to estimate relationships between the clones. And here I'm doing a comparison between the marker relationships and the pedigree relationships. There is an overall correlation of 0.63, not that high, between those estimates. And we are able to detect some outlier clones that we decided to not use in the following analysis. Basically, they look like replicated individuals. Maybe there's some mislabeling. And then this comparison allowed us to do this filtering. Yeah, basically those are 17 pairs. Okay, so I'm going to show the first results that are genome prediction for clonal prediction. So let me just explain what we did. So basically, we did a two-step analysis. In the first step, we got blues based on the raw phenotypic records, and then use those blues on the second step as our phenotype to do the predictions together with the markers and pedigree information. We are using here only data from the stage 3. Here are the linear model that I used on the first step. Basically, I want to point out that we had a term for the clones, and also we controlled for spatial relationships between the plants on the field. On the second stage, then you had the linear model with two random effects. One that's correlated using the relationship derived from pedigrees, and another from the markers. And I'm going to do a comparison later in which I use one term or the other. And here's how we're doing our validation of the predictions. So basically, there are going to be two ways to compare. One in which I'm doing cross-validation with clones from the cycle 05, which is a larger data set. And then, so basically splitting the data in 10 folds with 6 replicates. In another approach, we're going to use the whole data set from cycle 05 to predict the values from cycle 06. So here, before showing the results, I'm going to just presenting some heritability, broad sense heritability estimates from the traits that I'm considering here. And I'm going to also use this to talk a little more about the traits that I have. So basically, we have the ones that I mentioned, bricks, fiber, pole, and weight. But they were measured in some combinations of conditions that are different. So we have a first harvest and a second harvest for the stage 3 data set. And also we have the 2 cycles that I mentioned. And we also have 2 seasons. So in both cases, half the population were phenotyped on the beginning of the season. That's the early set. And then another half of the population in the end of the season. That's the late season. That's why we have so many combinations of the traits, and then I basically consider them as individual traits in this analysis. So here's the first results from cross-validation. Here, then we have the 4 traits: bricks, fiber, pole, and weight, and harvest the first and the second harvest. And in these bars, we have the both early and late trait. And you can see here that we have overall prediction accuracies ranging from a little above 0.1 to close to 0.4. And there is actually not a clear trend between harvest 1 and harvest 2 and between early and late. I cannot say that I can always predict better for one or the other. And between the traits themselves, in this particular case, there's a little difference between them, but we can see that fiber is a bit better than weight. In this cross-validation, basically separating the early and late sets, but another analysis that we can do is to put them together. That's what I'm showing here. So basically, by doing this, I increase the training sets. I have a larger population to do my training, and then I can do prediction to both the same the individual sets, early or late, or then to predict them together. In some cases, there was an increase of prediction accuracy, but there's not a clear trend for all of them. Especially in this case, it looks like there was a good increase in prediction accuracy. And in these predictions, I'm using both the molecular marker and pedigree in the model. So I have that full model with the 2 random effects. And here I'm comparing what I get when predicting only using the molecular markers, and then here only using the pedigree information. So basically the dots here are the same traits that I was showing on the previous plot, but now the values are the accuracies themselves. And then we see that there is a trend of higher accuracies when using the markers in comparison to the pedigree. And between this plot on the here and this one, basically the difference is that I include the previous result here with markers and pedigree and compare with only the pedigree. So they look very similar, which means that basically what's driving the increase in accuracy here is the prediction from the markers. So let's move to the other scheme of validating the genome prediction, in which I'm basically using the whole cycle 05 to predict the whole cycle 06. The same. So here we see a drop in prediction accuracies for all of the cases in comparison to what we had before. And the same idea here holds. There's not a clear trend between the harvest and the early and late seasons. And we can also include the early and late seasons together for prediction. In this case, we get some cases, especially here in fiber, where there is a bump in the prediction accuracy. And in some cases, actually, a big drop. The same comparison here now between prediction using markers and pedigree. Now then it's not super clear a distinction between, there's not a clear advantage for using the markers in this comparison. So some final remarks that are between these analyses. So there was the first paper that reported genome prediction sugarcane was by Gui et al. In 2013. They actually had higher accuracies for bricks and stock number, which are the two traits that we have in common here. Actually, I don't have stock number, but stock number is reported to be very correlated to weight. So maybe you can trace a comparison in this case. They had similar heritabilities, but then higher accuracies. And there's one important difference between their study and this one, which is the population composition. So in their study, they were using varieties and clones derived from breeding programs all over the world. And that, I think that's the main difference between their study and this one. Here, we were doing a study based on clones derived from an actual breeding program during the selection. And I think that can be something to keep in mind when thinking about implementing genome prediction in a breeding program for sugarcane. So some other comments. So on regard to the prediction across cycles, actually we can see that there's a lot of GBI. So maybe that's not the best way to compare. That's expected to be a drop in the prediction accuracy. In the same way here, it was interesting to do this comparison because it kind of looks like what we might observe when we first deploy sugarcane in a breeding program, if we also do the phenotyping of the first selections. I think if you had multiple year data, maybe could get some improvements in our predictions and get a better, clear view of the trends with regard to the GBI that's happening. And then with regard to whether to use markers or the pedigree for prediction, we could observe an advantage in doing cross-validation. And maybe this points to that if you use these predictions to select within families, then you would have a really clear advantage for using the markers. In that case, we cannot do the prediction for using the pedigree. Okay, move on to another topic, which is focused on predicting parental breeding values. So basically, I'm using the same data set as before, but instead of predicting the clone performance as I did, I'll look to the parents from that derived those clones in the breeding program. And I'll estimate breeding values for them and also do prediction. So in this case, we're going to do a single-step analysis with all the records that I have. And I'm going to put together data from stage 1 and stage 3. In that case, we're going to have a kind of very unbalanced data set in which we have parents that have from 2 to more than 200 records available in this case. So the breeding values are going to be derived from a very unbalanced data set. And also, because I don't have markers on the parents for this data set, I'm going to use the approach by Legara 2009, which uses what we call the H matrix, which is a combination between the pedigree relationship matrix and the marker relationship matrix. Basically, we can expand some of the marker information to the whole matrix using this approach. And just as a comparison here, when I correlated the pedigree matrix to the H matrix only on the set of clones that don't have marker information, we get a correlation of 0.73. If you remember before, we had a correlation of 0.63. So they're still not super highly correlated. So there is, and this kind of shows how one information pushed the other. So here's my linear mixed model. There's a bunch of ladders here, but what I want to focus is on the term G, which is the random effect for the genotype. And then this random effect will use this H matrix that I'm talking about. Actually, sometimes I'm going to use the H matrix, sometimes I'm going to use the pedigree matrix. So I'm going to do prediction using the H matrix and then compare to the pedigree, to the breeding values derived from the A matrix. In this case, I'm going to do prediction across cycles again, using all records from 2005 to form the training set. And I use the parents that are only in 2006 as my validation data sets. So here's the results. So basically, we have predictions in which I'm using the pedigree marker relationship, the pedigree relationships, the H matrix, and also an HW matrix that I'm calling here, in which I form the subset of the data for which I have both marker and pedigree information with both matrices. And then in this case, there was a higher prediction accuracy for weight, but in general, there was very little increase for using the marker information, and even a decrease when using weight. Another comparison that I did here was to include in the observed data the previous cycle, which is kind of I'm kind of reusing the same data that I'm using as training, but that's what brings the best estimates for the breeding values. And then there is a big increase in prediction accuracy here. I would say that actually the real prediction accuracy that we might expect is not this high. It should be something here a little lower. But I'd like to show these results so to have another information in mind. Another comparison that we did was to look to the parents and see if there are some full SIBs there. So basically, we had a couple of families in this data set, in the parents data set, with an average of a bit more than 5 clones per family. And we did the same estimates of correlation that we did before, but now only looking to those full SIB families. And in this case, we can still do a good prediction for plot weight, but there is a sharp decrease for bricks. I tested actually the significance of this. As you might imagine, there might be a lot of variation for this small number of families. And actually, the standard deviations here are pretty high. So if I do a t-test, it's not gonna be significant. So it'll be interesting to do this analysis again with a larger number of families or larger families. Some remarks here. So in this case, one interesting thing was to use the progeny data to do our predictions in comparison to what we did before, which was the data derived from the clones themselves. And so by doing that, we were able to combine data from many stages and multiple years. And also the mixed models allow to despite of that, to analyze these very unbalanced data sets. In a comparison between markers and pedigree prediction, we can see that again, we have probably a better use of the marker information when you look to prediction within families. And also in this analysis, we could use this H matrix, which kind of make it easier to use to access information that we might already have in the breeding program, but for which we don't have marker information yet. Okay, so in this last section, I'm going to talk about something a little different. So I said in the beginning that sugarcane is a polyploid species, and I was only looking to the markers in a very simple way, only looking to the presence or absence of the marker of the alleles there. So actually, we know that we might have more information there, right, because it's a polyploid. So in this section, I'd like to explore a bit more on the markers. So this is a diagram that's pretty common to see in sugarcane presentations that kind of shows how the chromosomes in sugarcane look like. And so here they're putting the colors to each of the species that forms the hybrids. And then the main thing that I'd like to point here is that, yeah, we have many copies from each of the chromosomes. So that's why it's polyploid. And also we have things like aneuploidy. So we have different numbers of ploid levels for each chromosome. And actually, this might happen even between clones. So if I go to another clone, I should not expect that these numbers for the same chromosomes to be the same. So it kind of makes things a bit complicated here. So here I'm showing a bit more on detail on what I mean by this, by what's happening to the markers. So it might happen because sugarcane not only is a polyploid but an autopolyploid, that we can observe markers in higher dosage. So we might have things that we observe in a diploid, like a single copy or absence, but also higher dosage of the alleles. And basically, that's the kind of difference that I'm missing in my previous analysis. It turns out that's one question that we might have is, okay, does it matter, right? Does it matter this dosage? It might happen that, like, after a dosage of 0, like when I compare to my traits of interest, an increase the presence of the allele in more copies doesn't matter anymore. It might happen that it really matters, that as I get more higher dosage, I have a higher impact on my trait, or something crazy might happen. Don't know, right? So basically, my point is, if we don't look to the dosage, we are not having the chance to look to this. Okay. So is it easy to look to that? And there are reports in the literature that explore this idea of looking to the marker information that you get from a population and try to identify in sugarcane what's the dosage, even in the case of alleles that appear in higher dosage. it turns out that it's kind of hard. There are a lot of cases that we kind of don't see a clear result. They confuse a lot. And then here I'm kind of summarizing what I think are the problems in looking to the higher dosage in sugarcane. So if you're doing an analysis of markers for dosage in sugarcane, first of all, we need to know what's the dosage that we have on a given locus. And or, I mean, the maximum dosage or the ploid level. But we don't know this information yet. And maybe we could have an approach that through the analysis gives this ploid level. That's another possibility, but it's hard to do. Another thing is that sugarcane is highly heterozygous species, and as other plants, we might observe a lot of duplications of genes and so on. So it might happen that the signal that we're observing here, I'm using signal to mean the molecular marker information that we get, be it, for instance, in our case, the sequencing information from locus. We want that signal to be really derived from every single allele that we observe on the locus. But due to heterozygosity, that might not happen. Or you actually might get signal from more than one loci together when we are clustering the sequencing information. So there are some difficulties here. And another is actually that we want the signal information, the reads that we get from sequencing in our case, to the number of reads to be a function of the dosage. And for instance, if we have narrow read sequencing, that might be a problem. Due to sampling errors, we might get a different number of reads than what we would expect from the dosage itself. So this is challenging. So what I'm going to do here is to try to avoid some of these questions. So I'm going to do an analysis that simplifies how we look to the marker data, which is to use the signal directly without doing any of those fancy statistics analysis on the marker data in which we find the dosage itself. I'm going to show examples of this later. But I'm going to use that so we can test this question that I made a few minutes ago. Is it worth it to use the dosage information when doing quantitative analysis in sugarcane? Maybe it's not worth it, right? So maybe a simpler analysis in which we just look to the presence and absence of alleles, that's enough. And that we can't do better than that. So I'd like to test this hypothesis. And another thing is, okay, we do all this analysis trying to find the dosage of, like as I showed in the literature, of finding the dosage information from the markers. And maybe if we can test that the use of this information is valuable, then it will give more incentives to follow on that kind of analysis. And we are going to do this through both GWAS and genome prediction. So building on the previous results, we're going to do an analysis in which I have the markers on a simpler representation, and another in which that doesn't take into account the dosage information, and another that does, and compare the results. Oops. So basically, I'm going to reuse the information that I got before, that we had before. And here, I'm going to explain what I mean by another representation of the markers. So I'm showing in this table 4 data points, 4 SNPs. And here I have read counts from both of those alleles from these SNPs. So in one case, for instance, I have read counts from my reference allele, but not from my alternative allele. And in my, what I call discrete genotype or discrete coding of the marker information, I'll assign a value, let's say 0 to this case. And when I have both alleles, I'm going to say it's a 1. And when I don't have one of the other allele, I'll say it's 2. That's basically what I did before. Right? But in this new approach, what I call a continuous coding of the genotype, I'll basically get the ratio between the read counts from the reference allele to the total number of reads that I have. So I will have something that's continuous. So that goes from 0 to 1, and that allows me to distinguish cases like this that might have, and the assumption is that if I have a higher dosage, I'll have higher read counts for that allele. So using this assumption, we build this continuous genotype for the marker function. So here's an example of how that works in the actual data that we have. So I'm showing here one SNP and another SNP. This SNP, on top you have the continuous coding, and on bottom the discrete coding, and those are the like histograms from my I had under discrete coding just the values 1 and 2, for instance. But in the continuous coding, this value, that's the heterozygous, they are, let's say, in a range of values that go from 0 to 1. And this is kind of flipped. So these guys here appear here. Okay. And this might be interesting because then you get a higher resolution for this. It also gives access to information that we didn't have before. I'm showing here one SNP that only had one. So if you're working with a deep learning, expect this to happen. Right? Only have heterozygous individuals in your population. But in sugarcane, that happen. At least it happens in my data set. And, basically, I throw out markers like this in a discrete analysis. Right? But in this continuous one, I actually can distinguish that between one and another. And so what I'm going to do, kind of markers, I'm going to do I'll have two matrices, what I'm calling D from discrete and C from continuous, with markers in both representations. And from the markers that are like this, that are marked on discrete, so with extra markers that I didn't have access to before. Before just filtering data. So I had some samples that are replicated. So I did an analysis taking into account the Euclidean pairs of varieties that are replicated and non-replicated, and compared those two distances to find an optimal read depth for filtering these SNPs. So the optimal one is actually 80. I used some 50 just to not lose that many SNPs, but it's still 50 to get basically non-significant difference here. So using those HCs, the D, C, and C2, I did a PCA analysis from the data set. And basically the same, showing here the same stage 3 clones that I used. And we see that using discrete coding and continuous, the same markers, I get a very similar. Actually, we have 0.92 correlation with the off-diagonal elements in those matrices. The relationship is derived from the. If I look to the two matrix, I actually have a lower correlation between them. And in the first PC, in the second PC, we kind of see slightly different. It looks like we have some extra information that we didn't have access to before that we're gaining here. So let's go to the actual tasks with this through coding. So I'm gonna do. So okay. So first the quick plots from the JAWS analysis. So I have. So here I'm comparing the continuous coding and the discrete coding, and there's not a super clear difference between them here. I'm just showing that I might have more significant results on these traits harvest too early. And when I do a Manhattan plot, it becomes very messy, difficult to see a clear trend. So I try to compare actually the continuous and discrete coding in a slightly different manner, which is going over a range of thresholds instead of just using X1. For a range of thresholds, I count the number of significant hits that I have for that threshold. And then here in this plot, I'm showing the number of hits that we get. And on red, when using the discrete coding, and on C, on green, using the continuous coding. So we get a higher number of hits when using the continuous coding. That's good. Right? And well, actually, if you do this analysis here, here I'm putting together C and C2, so it's not that fair for the discrete coding. I have more markers in this case for the continuous coding. But for in this case, I just compare the C and the D matrix, and still we have a higher number of hits in that case. So I can look to both cases. In one case, we're getting more markers due to the use of the C2 matrix, that markers that are not polymorphic in the discrete coding. But even if I just focus on the markers I have available already, I still have an increase in the number of hits. So in order to compare the modeling using the linear model and the GAM model, I also did the same range of thresholds, but then I just checked for markers that are not significant for linear modeling. How many of them are significant for the GAM modeling? Basically, because of the likelihood is not directly comparable between both models. So I see here an increase in the number of hits, between 6 and 15% more in comparison to the number of markers that we get from only using linear modeling. So we also did genome prediction. In this case, I used the GBLUP modeling, but also some other ways of modeling the data, RKHS, support vector machines, and random forests. So those are the results for genomic BLUP in the 4 traits and the different matrices. So let's focus on this one. So in most of the cases, there are a slight improvement in using the continuous coding. And even when I use the C2 matrix, which has, I didn't show the numbers here, but my C2 matrix has only, so here we're talking about around 50,000 markers. The C2 matrix has 5,000 markers. So we still have a high prediction accuracy in comparison to the D matrix. And well, not always, but in general, there is a little increase in prediction accuracy. To include the other models for genome prediction, I don't observe a real improvement between the different models, and sometimes it gets really worse. So I'm going to stick with the GBLUP results here. I also did a multi-kernel analysis in which I combined the D matrix with the C matrix into random effects. And that didn't, I'm not going to show here, but it didn't improve much. So some conclusions here. So the use of this continuous coding actually provided some gain in information. So I have more markers that were not polymorphic before. And also, because of the relationships were a little different, it looks like we're getting access to new information that we didn't have before. In the actual tests in GWAS, we had more hits. Genome prediction, it was about the same. So it's not clear whether it was a real improvement in accuracy there. But I think we can say that to use some forms of dosage encoding in studies for sugarcane might be beneficial. So that's what we are actually investigating. So just to finish up, I'd like to thank some people. So especially, so all this study was my PhD, the whole program, the data, everything comes from CTC. And we had actually a lot of people there that contributed to get the data. You can imagine the field records and everything. But Karine, Francisco, and Thiago, they were directly involved in establishing the framework for this project. And also Michael Butterfield and William Burkins, they were responsible for actually sponsoring everything here. Also, I'd like to thank a lot my special committee, Mark, Jean-Luc, Jean Booth, especially Mark for accepting me here and guiding through all those years and supporting every step. And also, I'd like to thank a lot all the Soros and Genomic team. We interacted a lot through those years. I learned a lot through that. Here I'm bringing this old picture because I think it has some folks that are not here anymore. I'd like to especially mention Denise Akdemer, Nick San Antonio, and Uche, it's not here, that gave a lot of input to my analysis. So that's it for today.
Running a little long, but we'll take a question or two.
I had a question about, so your prediction between cycles. You talked about the common parents. There were a subset that were used in both cycles. So I'm wondering if just from those original predictions you did, if you ever look at the accuracy on the cycle you were predicting just for the common parents, because you should be able to predict within family better than just across the whole group.
So you're suggesting to use the parents that I had in common. I think it'll be
just to look at the accuracy was what I was thinking. You use the whole training population to do the prediction, but then maybe you're predicting so that when you correlate with the whole group, that's what you showed. But just look in those common.
I actually did that. It's just not showing. There is a higher accuracy when I do that.
Okay.
It's not like super high, but yeah, it's higher than that, significantly higher. I should have included that.
Is there a concern between when you look at your dosage of an allele versus the difference in a read depth of a region?
There is a.
If there's a, are you looking at a difference in whether or not you have a read depth or whether or not you have a difference in dosage in your reference versus something?
So ideally, we would be looking to the dosage, right? That's what we actually have in the plants. It's the dosage, not the read counts. So the thing is, we don't have access to that information yet. Right? I'm not sure if we're going to get a date when we have in sugarcane. So I'm trying to here actually not propose that we should use the read depth as a tool. And I think we still need to find a way to get the actual dosage. But what I tried to do was to try to ask a question. Okay, is it worth it to look at that? Should we do that? Because actually, I had this question when I got here, and that's why all my initial analyses are based only on the presence or absence of markers. And if I went to do a actual dosage analysis, probably I would be working with way fewer markers than I have now, and actually it's super complicated to do that. So actually not proposing to use this kind of way, but can we still answer some questions by using another way to look at the markers?
I hate to cut this off, but we've got to get back out there and harvest mulberry. We're taking volunteers, by the way.
This has been a production of Cornell University, on the web at cornell.edu.